Hello, everyone. Welcome to today's live broadcast, Challenges and New Solutions for Isolating Exosomes, Other Cellular Vesicles, and Vesicular RNAs. I am Rachel Brown of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN. I know you will all agree that the pursuit of any biological research begins with a sample. It can be blood or tissue or cell. As you all know, QIAGEN offers you innovative solutions that cover every single step along this journey, not just technologies, but bridges from samples to insights. For more information, please visit QIAGEN.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Dr. Martin Schlumpberger. Dr. Schlumpberger has a PhD in chemistry and more than 20 years of experience working in genetics and molecular biology. He did his postdoctoral fellowship at the Institute of Neurodegenerative Diseases at UCSF. He has been at QIAGEN for over 13 years developing new strategies and products, both manual and automated, to isolate nucleic acids from a wide variety of samples, including extracellular RNA and miRNA from serum and plasma, exosomal RNA, as well as intact exomes and other extracellular vesicles. He started as a research scientist and is currently an associate director in the Sample Technology Development Group. I will now turn it over to Dr. Schlumpberger for his presentation.
Hi, everybody. Thanks for listening in and for your interest in this topic, and also for LabRoots for inviting us. We seem to have some network problems, actually, so I will ask to have the slides advanced, and I hope there won't be any other issues. So with that, I go on to the first slide. This is just a visual impression of what we're talking about. So on the left hand is shown a scanning electron micrograph that shows a glioblastoma brain cancer cell. And you can see it has these protrusions and budding vesicles all over the surface. So this is just an example of how, for many tumor cells, basically produce these extracellular vesicles in really large amounts. And on the right-hand side, you see a transmission electron micrograph showing what is called multivesicular bodies. This is part of the endosomal pathway, where basically vesicles pinch off from the endosomal membrane, and so that there are generated intra-endosomal vesicles that contain cytoplasmic material. This is what the two arrows show like. And then these multivesicular bodies can fuse with the plasma membrane and thereby release their vesicular content into the surrounding media. That is also shown on the next slide in a schematic, I think. So this is slide 3 now. And it basically shows again in the lower panel here the schematic drawing of the multivesicular bodies, how they originate by endocytosis. Then the intra-endosomal vesicles are formed and then finally released when the endosome fuses with the plasma membrane. And this is the canonical pathway for what most people call exosomes. There is unfortunately, some discrepancy in how the definitions are being used, but most people define exosomes via these pathways. And exosomes are usually in the size range of about 30 to 100 or maybe 150 nanometer. Again, depends on who you are talking to. And then extracellular vesicles can also pinch off directly from the plasma membrane. This is shown in the second illustration that's right behind the other one, or on top of it. And vesicles generated that way are usually a bit larger than what I just said, around 200 or 150 to 300 nanometers typically. And these are often referred to as microvesicles. But like I already said, the definitions overlap and some people use the terms interchangeably even. So why are we interested in looking at the content of those vesicles? The reason is, as you have seen in the first slide, for example, cancer cells are producing lots of these vesicles, and so what has been shown is that they actually contain a snapshot of what's going on in the tumor cell. And so we can use that information, and that is shown on slide 4. We can use, for example, not just exosomal RNA, but also other exosomal content, of course. I'm going to be speaking mostly about RNA, but obviously protein and other biomolecules are also of interest. So basically, these vesicles are found in all kinds of body fluids, mainly blood, urine, but also CSF, saliva, and tears, breast milk, ascites, synovial fluid, pretty much anything you can think of. And those exosomes, like I already indicated, contain RNA. And what is nice about it, the vesicular membrane actually protects these RNAs from RNases that are present in biofluids, especially plasma is full of RNases. And any naked RNA is basically completely degraded within seconds. And what is inside the vesicles is safe and stable, as I'm going to show. And that RNA actually provides a snapshot of the transcriptomes of the cells where they come from. That could be tumors, metastases, and also other kinds of diseases like, liver cirrhosis or toxicity. All kinds of cells basically are producing these vesicles, and diseased or stressed cells typically tend to produce even more, which is nice if you want to get to use this for figuring out what is happening in the organism actually, or what is going wrong. And there are actually two ways you can use this, especially with regard to cancer. You can obviously profile RNA levels and use that expression profile as like a fingerprint of what is going on also in for example, tumor cell. And the other thing that you can also use the RNA or also DNA for is to look for disease-specific mutations. Again, that's relevant particularly for cancer, where we can look for people have detected KRAS, BRAF mutations and all that in the RNA that is basically released into the bloodstream. Next slide. So from this, I think it's pretty clear why people are so interested in those vesicles and characterization of the vesicular content. So with respect to RNA specifically, there are some important considerations that you have to have in mind when you start doing experiments like that. And I think pretty much the most important is the background from cellular RNA. Obviously, blood contains lots of blood cells, and the RNA content in cells is several orders of magnitudes higher than what is found in vesicles, mainly, or if only by their much larger size. So you have to be very careful to exclude any remaining cells from your starting sample, be it plasma, serum, or saliva, because if you don't do it, you will essentially have cellular RNA blanking out your extracellular signal, just because it's more abundant. And in our protocols, or also what most people recommend, is to do an upfront centrifugation or filtration step in addition to what is usually done to generate plasma, to make sure there is really no cellular fragments or apoptotic bodies and thrombocyte fragments remaining. Another consideration is specificity. Because there is, I just said that cell-free RNA is usually degraded when it's in plasma because of the RNases. But there is at least a population of cell-free microRNA that is associated with proteins and thereby protected from RNases. So you do have a certain population of non-vesicular cell-free RNA, and depending on what method you use, this is most methods will not distinguish between these two populations. Like if you do ultracentrifugation to pellet extracellular vesicles, you will also pellet larger protein complexes to some degree. And what we did, the protocol we designed actually is specific for the vesicle and vesicular content. And so this allows people to distinguish what is in vesicles and what not. And I guess the question is still open in which areas this is especially useful. But what you can imagine is cell-free RNA that is not in vesicles is probably mainly released when cells die, apoptose or necrose, and then the active RISC complex is basically released, and the AGO2 protein that is associated with microRNAs protects them from RNases. And on the other hand, the production of vesicles is an active process, and so you could easily imagine that depending on what phenotype or disease you're looking at, it may lead to generation of free RNA by necrosis or apoptosis, or maybe some other phenotype or disease will mainly result in production of vesicles. So if you have seen the picture of the glioblastoma cell in the beginning, you can easily imagine that in case of cancer, a lot of what is released will be in vesicles. RNA purity is obviously also of concern, and so here it's mainly important to have a protocol that produces really high quality RNA and also intact RNA. And I will show you some data on how intact the RNA is that we can get from vesicles. And finally, a consideration is also how much sample can you actually process, how much sample you have available, obviously, and how much you can process. And so the issue is that many transcripts are really present at very low copy numbers in the cell-free fraction. And that's especially true for messenger RNAs. MicroRNAs are usually present at higher copy numbers, and messenger RNAs not so much. So especially if you're looking at messenger RNAs and mutations, for example, you want to process large volumes of sample to get the required sensitivity. And so what I'm showing, the method is able to deal with up to 4 ml of serum or plasma. And with that, on the next slide, this is just shows how the basic, how the method works basically. This is a flow chart. Like I already mentioned, after we generate the plasma by centrifugation, we employ an extra filtration step to eliminate cell fragments, apoptotic bodies, and the like. We recommend to use a 0.8 micrometer filter for that because we think that is the right cutoff to make sure that there are no cell fragments. In principle, one can also use centrifugation and change the conditions or the filter pore size a bit if the interest is mainly towards smaller size vesicles. You can spin it a bit harder beforehand or use a smaller pore size. The filtered plasma is then mixed with a binding buffer, applied to a spin column, and here we use a membrane that retains all the vesicles that are present in the sample. So we didn't really think it would be useful to go after particular subfractions of vesicles at this point, especially considering that there's no agreement really in the field which population or subfraction of the vesicles is really the most important. Depending on what people are working on and what methods they use, everybody prefers a different subfraction, and so this method will give you all or the entire vesicle population, everything that is smaller than the filter pore size that you employ. Then we wash, and we actually, when we go for RNA, we do a combined lysis and elution step, and then so we take that lysate and apply it to an RNA column to isolate the RNA directly. So we don't isolate vesicles first, we bind them to the membrane and then destroy them in the elution process. This is what we call the exoRNeasy kit that is also mentioned in the title. We also worked on a protocol to release intact vesicles. This is basically in the middle column in the lowest panel where it says lyse vesicles. So at this point, we have a different version of the same protocol or also the kit where you can use an aqueous buffer to release intact vesicles. And then those vesicles can be used for physical characterization, sizing, or also for biological experiments when you can isolate vesicles and then apply them to, for example, cultured cells or even to animals and see what changes or what phenotype they cause in the target cells. So that's, like I said, a different product that we are just releasing. On the next slide, this is basically an example of how the intact released vesicles look like. This is a scanning electron micrograph. So what you can see is on the right part, this is the eluate with intact vesicles that we get from the column. The size bar that is in the black space underneath the image, this is barely recognizable, I'm afraid, but the size bar you can just barely see there is 200 nanometers. This is in agreement with what is expected from the larger vesicles. And then you also see a lot of smaller vesicles that are in the size range of 30 to 100 nanometers roughly. And on the left panel, you can see for comparison, this is vesicles prepared by ultracentrifugation. We can also see some vesicles, just similar to what we see in the exoEasy eluate. We also see a lot of smaller particulate matter in the background, which we think is probably larger protein complexes that are co-pelleting in the ultracentrifugation protocol. And we get less of that in our method because the membrane basically selects for the vesicles. Next slide. We are on slide 8 now. And this basically is supposed to show that we get nicely linear recovery of RNA dependent on how much sample input volume we use. Here we are starting at 500 microliters and up to 4 ml. You can see that the CT values for a number of different transcripts that were analyzed here decrease in a linear fashion, about roughly 1 CT value, 1 cycle for each doubling of the sample volume as expected. So there is a few messenger RNAs here like BRAF and HPRT, and 2 microRNAs, LED7A and miR16, and ribosomal RNA that we also detect in the vesicles. And so 4 ml is the nominal binding capacity of the column for serum and plasma. And you can see if we apply larger volumes to the column, the CT values do not improve much any further and may actually decline a bit. But up to that, it's perfectly linear. Next slide. Slide 9 now. So the question is, what does the RNA actually look like? And so here's a representative electropherogram from the Bioanalyzer. And we comparing here exoRNeasy eluate, so compared to RNA isolated from ultracentrifugation pellet. In this case, from 2 milliliter of plasma. And so what you can see, most of the RNA is actually pretty small, around 100 base pairs or less. There is some larger RNA, but not terribly much. So it looks very, very different from what you isolate from cells. And for most people, electropherogram pattern like this indicates that it's really just degraded RNA. And in fact, that's not really the case, as I can show you in the next couple of slides. So the next slide, basically, this is still the Bioanalyzer. And what we did here is we took we fractionated the RNA we isolate from the vesicles into a small and large RNA fraction by modifying the ethanol used for the binding step. And so you can see on the left side the small RNA fraction. You see again that relatively large peak of small RNA. This is microRNA, but also other stuff, tRNA and some other small non-coding RNAs. And on the right side, you see the large RNA fraction, and you can even still see some ribosomal bands. They're not nearly as prominent as in cellular RNA, but they're still there. And some other large RNA. And from the ribosomal bands, I think this is the first indication that the RNA is actually not really degraded, because we don't see the ribosomal bands smearing towards smaller sizes. So we looked at some messenger RNAs, whether they are recovered intact from the vesicles or not, or how intact they are. And we did that by PCR. This is shown on the next slide, slide 11. So for this experiment, we basically isolated RNA again from the extracellular vesicles. Then we prepared cDNA using 2 different methods. In one case, we used random hexamers for cDNA synthesis priming, and in the other case, we used only oligo-DT. So if the RNA is degraded, the further away your PCR amplicon is from the poly-A tail, the higher or the lower the chances are that the cDNA actually extends that far because of strand breaks. And what we would see then is that the CT values are higher than they are with the random priming strategy. And as you can see in this example, the CT values are all pretty much the same, no matter whether we use oligo-DT or random hexamers, even as far out as 5 kilobases from the poly-A tail in case of the KRAS amplicon. So this indicates even up to 5 kb out the RNA, there is no sign of any degradation, which is on first glance, it's pretty surprising if you consider how the electropherograms look like. On second thought, I guess this is illustrates what I said previously, that when the RNA is inside the vesicles, it's really nicely protected from everything that is outside. So this is like a little treasure vault that keeps your RNA safe. And obviously this makes it even more useful for basically use as biomarkers. On the next slide, what we're looking at here, we are comparing the RNA we isolate from the vesicles, from the fraction that binds to our membrane, and the fraction that does not bind to our membrane. So we took the flow-through, the column flow-through from the binding step, and we isolated RNA from that column flow-through, as well as from the fraction that is retained on the membrane. And then we did a couple of, or a number of qPCR assays. First of all, the lighter shade diamonds are 4 different microRNAs. And for this analysis, we looked at microRNAs that were previously described in a paper that is referenced here below the graph by Arroyo et al. They did some profiling of microRNAs that are in vesicles or not in vesicles in plasma. And we choose 2 that, according to that study, are primarily outside of vesicles, miR16 and miR92A, and 2 other microRNAs that were reported to be mainly inside vesicles. Let's have an A and miR. There is a typo here. It should be miR142-3P. Sorry for that. And when you compare the CT values, you can see, sure enough, the blue and red ones, miR16 and miR92A, we get similar or even slightly lower CT values when we analyze the flow-through, indicating that a large part of this is actually not in vesicles and not retained by our membrane. And then if you look at the, let's have an A and miR42 signals, the miR42 is actually underneath the miR16, so it's not really well visible in the left column. But you can easily see that the CT values are much higher in the flow-through, indicating that most of that is in vesicles and retained on the membrane. Now the remaining data points are for messenger RNA. So we have BRAF, KRAS, HPRT1, a ribosomal protein, and GAPDH. And we all detect those, some of them just barely, but we can detect them in the vesicular fraction, but we do not detect pretty much anything at all in the flow-through, indicating that everything is retained and presumably all within vesicles. And this actually makes sense because in contrast to the microRNAs that can be protected by a protein like AGO2, it's much more difficult. I don't think there is a protein really large enough to fully protect a long messenger RNA the way microRNAs are protected by AGO2. And I think that's what is reflected here. And what it also shows is that the vesicles are retained very efficiently on the membrane, because if we would bind only 80% or something like that, then we would see, then we would still see stuff in the flow-through at only about 3 cycles difference. But as you can see, the difference is much larger for the messenger RNAs. So we have a really good binding efficiency on the membrane. And so you may ask, how do we know that this, what is retained on the membrane, is really inside vesicles and not retained for other reasons? And to actually demonstrate that, that is shown on the next slide. So we are on slide 13 now. Here we did an RNase protection assay. Basically, we followed the protocol. We bind the vesicles to the membrane, and then we apply to the vesicles while they are still bound to the column membrane. For the rightmost group of columns, we did not add anything. And for the second group from the left, we added only RNAs. And you can see that the signal doesn't change terribly much. So even though we add RNA to whatever is bound on the membrane, the RNA is not able to degrade the RNA. Only if we add tritone as well, this is the group of columns to the left. When we add RNAs together with tritone, you can see that the RNA is pretty much entirely gone, destroyed by the RNAs. And so the tritone indicates that this is a membrane that protects the RNA from the RNA, so vesicular membrane. And if we add tritone alone, we do see some degradation. This is presumably due to some RNAs that is still present on the membrane that has been carried over from the plasma sample at this point. So I think this shows very nicely that the membrane, the RNA we isolate from the membrane is protected by vesicular membrane. On the next slide, this is basically what I said previously. This is regarding sensitivity. Here we looked at how much messenger RNA can we actually detect in the cell-free fraction from plasma. And what we used here is what we call a PCR array. This is also a product provided by QIAGEN, obviously, that has 96 or 384 different messenger RNAs. In this case, it's a 96-well array. So we have actually 84 different messenger RNAs and a bunch of controls. All the assays are pre-arrayed on a plate, and we only add the sample and the master mix to that. And then we can basically profile that set of messenger RNAs. In this case, it was a set of known oncogenes. We actually included a pre-amplification step to make sure we get enough material for all 96 wells. And what you can see, or what the diagram is illustrating, is when we start with 200 microliter of plasma. This is the lowest box in that box plot. From 200 microliters, even with the pre-amplification, we detect only about half of the messenger RNAs arrayed on that plate. And when we go from 200 microliters to 1, 2, and 4 ml, we detect progressively more of these transcripts, and at 4 ml, we basically detect almost all of them. So this shows, I think, pretty nicely the benefit of using a larger volume of sample. This is, like I said, especially true for messenger RNAs because they are not as abundant in the cell-free fraction as the microRNAs are. For microRNAs, on the next slide, slide 15, I have some data. This is also a similar PCR array. Now we have assays for microRNAs on the plate, and we profiled again the fraction that is isolated by the exoRNeasy protocol, by the column, and what is not retained on the column, the flow-through. And we compared the CT values. So on the x-axis is basically the abundance of the microRNAs. So to the right is the most abundant, to the left the least abundant. And on the y-axis, we show the distribution between vesicles and not in vesicles. So what is on the bottom is largely not in vesicles, and what is on top is mainly within vesicles. And marked in color are again the same 4 microRNAs that we picked from the literature and showed before on the previous, or one of the previous slides. So miR92A and miR16 are again in the fraction that is largely outside vesicles, and miR142 and LED7A are mainly inside vesicles. And what is also noteworthy, we also have here in the lower right corner, we have miR451, which is a marker of erythrocytes, so from blood cells, and so it's very abundant in blood cells and therefore also very abundant in plasma, and it's largely outside of vesicles, which I think basically reflects that this is there because of blood cell lysis, maybe during blood collection or extraction. Finally, on the next slide, I have an example for mutation detection. Here, plasma was isolated from a colorectal cancer patient carrying a KRAS mutation, and the isolated RNA was subjected to targeted RNA sequencing. And what you can see is we are able to detect the tumorigenic mutation in plasma in the cell-free portion. And actually, in this case, about 10% of all reads carried the mutant signature. This is also reflecting that tumor cells produce a lot of these vesicles. So that summarizes the first part of my talk. Conclusions: we have a protocol that extracts really high-quality RNA from extracellular vesicles. The vesicles contain small and large RNAs, and the large RNAs are actually surprisingly intact still. All of the RNA, pretty much all of the cell-free messenger RNA is in vesicles, whereas from the microRNA we have two distinct populations, inside and outside of vesicles. And by being able to separate these two populations of microRNAs, this may give us a better, more power in analyzing specific questions. And finally, the vesicles, we can detect the expression of tumor markers and also mutations, somatic mutations that are only present in the tumor. For the remaining few slides, I will just basically reflect on some questions that we've been asked a lot and that we did some follow-up experiments on basically. So the first, what we often, people are interested to isolate exosomes or exosomal RNA also from cell culture supernatant. Basically, cultured cells obviously also produce these vesicles, and these vesicles produced by cell culture can be used to answer a lot of questions. They have biological questions. They have also been used in some cases even for therapeutic purposes to deliver some cargo to other cells. We're also looking at the stability of the vesicles and lastly the influence of sample handling. So next slide, we're on slide 19. Now this is showing that the protocol works for cell culture medium. Here we tested different volumes of cell culture medium from 4 ml up to 32 ml. This is MCF7 cells that were cultivated for 48 hours, either in serum-free or vesicle-free medium. So obviously, since normal cell culture medium contains fetal calf serum or something similar, you have to make sure that you because the fetal calf serum, just like human serum, contains these vesicles, you have to make sure that you do not introduce this into your culture, because otherwise you will have a mixed population of vesicles from the cow and from your cells. So there are two ways to do that. Either you cultivate the cells without serum, or you use fetal calf serum that has been depleted of vesicles. You can buy that or prepare it yourself. And what you can see, the protocol obviously works for both. And we get a linear recovery of RNA from these cells. In this case, I think this is beta-actin that we monitored here, beta-actin messenger RNA. You can see we get fairly decent CT values, and the CT values decline linearly with the input volume. So in this case, up to 32 ml can be processed. Obviously, it will depend very strongly on what type of cell you use and the culture conditions. So this is only an example. If you don't know yet beforehand, we recommend to use not more than 16 ml. And I should also say that at 32 ml, we are actually at 4 column loading steps for the binding. I have some trouble with my connection right now. I can't see my presentation. Let's see, just okay. All right, so we can also, on the next slide, slide 19, we looked at how well protected is the RNA inside the vesicles. So what we did here is we generated plasma. We took one aliquot that we processed directly using the exoRNeasy protocol, and we took additional aliquots that we froze and then thawed once, or we actually did 5 freeze and thaw cycles also. And then we also did, as a negative control, we took one aliquot where we added SDS to destroy all vesicles. Actually, I'm sorry, this is cell culture medium, not plasma in this case. But still, we did either process the cell culture supernatant fresh, or 1 or 5 freeze thaw cycles, isolated the RNA, and analyzed it. And as you can see, we get identical CT values in all cases, so indicating that the vesicles survived the freeze and thaw cycles. And in contrast, if we add the detergent to destroy vesicles, the RNA is basically gone. So next slide, this is slide 21 now. This is actually done by a field test customer who, in this case, the customer isolated vesicles produced by HEC293 T cells, isolated the vesicles using exoEasy, so the protocol for intact vesicles. He did an additional buffer change and concentrating step by ultrafiltration over using a 100 kilodalton membrane, then labeled the isolated vesicles using a fluorescent lipid, and finally removed the excess unincorporated dye using size exclusion. And what you can see, the lower bottom, actually the labeled vesicles were then incubated with the target cells, again 293 T cells, a fresh culture, for 1 hour. And what you can see, the left is a negative control. This is dye without vesicles. There is no uptake. On the bottom is nuclear staining with DAPI. And on the right, we can see the uptake of dye into the cells. So the vesicles that we isolate are actually functional and are being taken up by cells. So, and for the last part, I will try to go over this briefly because we're actually short of time already. Here we showed how the handling affects your results. On this slide, we have different blood collection tubes, and we also have plasma and serum in comparison. And what you can see, first of all, on first glance, you see no matter what blood collection tube you use, we always get some results. We have a spike-in control here. This is the gray diamonds that are about CT value of 22, and you can see the CT value is very similar in all cases, which is indicating that we isolate and amplify this spike-in control with similar efficiency in all cases. The only thing that we not recommend, this is the second column here, is heparin, because heparin is difficult to remove and is a strong inhibitor of enzymatic reactions. And what you also see, if you look at different transcripts, that we do have some variability depending on what tube we use. And so the message here is you can use all these tubes except for heparin, but you shouldn't switch within a study. So if you do comparison between samples, you should always use the same type of blood collection tube. Next slide, we looked at, this is slide 23, we looked at what happens if there is a delay between blood collection and generation of the plasma. And what you can see is we looked at a range of different transcripts at directly after blood collection, and then after 1 day at room temperature, and after 3 days of room temperature. And then we compared the CT values. We took the delta CT between day 1 and day 0 and day 3 and day 0 for the bound fraction, the vesicular RNAs, and the flow through the non-vesicular RNAs. So the blue bars are the vesicular RNAs. You can see there is an increase after 1 day, about 1 cycle, so indicate that we have 2 times as much vesicular RNA, and after 3 days up to 8 times as much. And so what is important to keep in mind, this is vesicles that are produced by the blood cells during the storage. And what that means, most people are interested in the vesicles that come not from blood cells but from elsewhere, tumor for example. And so if you have your blood standing around, blood cells keep producing vesicles, this will dilute out your message that comes from elsewhere. So that's why it's important to avoid that. And the red bars show the flow through the non-vesicular RNA. You can see an increase after day 1 and then a strong decrease after 3 days. What we think is happening is that some of the cells die after blood collection, release RNA into the plasma, and then that is degraded slowly over time. Depending, like I said, the microRNAs are protected by proteins probably and survive for a while, and then they are eventually degraded. I think we can skip slide 24 for the sake of time and go directly to slide 25. This is the final data slide I was just showing here. Like I mentioned before, we recommend to do a filtration or centrifugation to get rid of blood cells, remaining blood cells, and cell fragments from plasma, and also thrombocytes or whatever may still be in there. And in this experiment, we basically analyzed the effect of different variations of that filtration or centrifugation step. First of all, the red bars you can see in the graph on the left. The red bars is plasma that is generated by standard protocol doing only a 5-minute 1900 G centrifugation. And when you compare that to plasma that was subjected to an additional 3000 G centrifugation or filtration on a 0.8 micrometer filter, this is the last, the dark purple bar, you can see that there is like at least 3 or 4 cycle difference. So that 3 or 4 cycles, that means basically a 10-fold difference. So 90% of the RNA that you see in the normal plasma is removed by the 0.8 micrometer filter. So 90% is in fragments or vesicles that are larger than probably cell fragments that are larger than 800 micrometers. So this is basically not what people are interested in usually. This is, like I said, apoptotic fragments and the like. And then I said we recommend 800 nanometers or 3000 G centrifugation, and you can see for all 4 transcripts that we tested here, the bars, this is the light blue and the dark purple, are very similar. So these 2 conditions gave roughly the same results in our hand. Whereas when you centrifuge at 16,000 G for 15 minutes, you can see that we lose some of the RNA, indicating that we get rid of some of the larger vesicles already. Yeah, and then we also when we combine 3000 G and 800 nanometers, we lose a little bit more. This is the dark blue bar here in the middle. So basically, I guess both protocols together or both treatments together remove a bit more than each one alone, even though they gave similar results each by themselves. And then just for curiosity, we also applied smaller pore size filters, 200 nanometers and 100 nanometers. These are the 2 remaining bars. And you can see that this further results in a further decrease in the RNA levels that we see. What I find interesting here is that for the 2 large transcripts, GAPDH or long transcripts, GAPDH and 18S, ribosomal RNA, that decrease is a lot more pronounced than in case of the 2 microRNAs, LET7A and miR16. And that indicates so that at the very least, the RNAs are not evenly distributed over the vesicles, and it also hints at the long RNAs being present in larger vesicles mainly, or more enriched in the larger vesicles, and microRNAs more enriched in the smaller vesicles. And we also looked at, from the same experiment, we also did one aliquot where we isolated intact vesicles, and we did physical characterization by nanosite, which analyzes particles by microscopic observation and Brownian motion, analysis of Brownian motion. And so you can see the size, the average size of the vesicles that we get is about 160 nanometers roughly, and that doesn't change much with any of these treatments, except when we get to a 100 nanometer pore size, the vesicle diameter drops. And similar for the particle concentration, only the 100 nanometer size, pore size gives a decrease, substantial decrease. So again, that also means that what is happening to the RNA is different than what we see on the vesicles, and again indicates that probably not all vesicles contain RNA. So conclusion, we can, of the second part, so we can isolate exosomes and exosomal RNA also from cell culture supernatant. The vesicles are very stable through several freeze-thaw cycles, and sample handling can severely affect your results. Choice of blood collection tube, a delay in processing of the blood, and also that the centrifugation or filtration you're doing is essential to remove the background of cellular RNA, but also affects, depending on what conditions you use, can affect the representation of different classes of vesicles and RNA. And the RNA is not evenly distributed, obviously. And then acknowledgements. So there are several people at QIAGEN, obviously, who contributed. And then I should say that the entire project, and also the product, was developed in cooperation with Exosome Diagnostics. And the data on uptake, vesicular uptake into cells, was provided by Casey McGuire at Mass General. Thank you for your attention.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, Bioanalyzer tray shows an increased amount of small RNAs in presumably the microRNA range compared to the exoKit samples. So how do you account for this abundance of the small RNAs given the highly variable EM images?
So can you repeat the question? What the difference was between which fractions? Or which preparations? I didn't.
Small RNAs in presumably the microRNA range compared to exoEasy kit samples. How do you account for this abundance of small RNAs given the highly variable EM images?
I don't get the question, I'm afraid. So comparing the exoEasy to what? We compare, I don't think I had any data like that. I'm just trying to figure out which, is that relating to a particular slide? And maybe we move to the next.
Perhaps you can ask the person who submitted the question to rephrase, and so we'll come back to that. How about?
Yeah, maybe I can also answer that by email later if necessary.
Okay, perfect. So the next question is, what is the maximum volume of samples that can be used with the kits for cell culture supernatant?
Yeah, like I said in that example on that was in the presentation, I think slide 19, we have tried up to 32 ml successfully, which is 4 binding steps. If for more than 4 binding steps, I think the protocol gets very tedious. And I think it depends also on what cell type you use. Some cell types produce really insane amounts of vesicles, and there the binding capacity will probably be reached a bit earlier. These were MCF7 cells, and so this is just an example. I don't think it's possible to really give an absolute number here in that case.
Okay, great. So our next question is, what is the expected RNA yield?
Yeah, in terms of RNA yield, obviously the amount of cell-free RNA is pretty low. From plasma, serum or plasma, we usually get about around 10 nanogram of RNA in total from like 2 ml of plasma or so. This is measured by the Bioanalyzer. The amount of RNA you get is, if you really exclude all the cells, is too low to be measured by, for example, the nanodrop or something. So the nanodrop may still give a signal from some impurities, but from RNA it's not possible. You have to use either estimate by Bioanalyzer or use something like ribogreen fluorescent assay to detect the nucleic acid from the vesicles. Okay,
great. So our next question is, how much plasma or serum would you recommend to isolate from in order to have enough RNA to perform next small RNA next generation sequencing successfully?
Yeah, so what I showed in my presentation, obviously those were examples mainly for PCR. For PCR, I can say that for 200 microliter, we can detect most microRNAs that are actually there. And for messenger RNA, I would use a lot more, like I showed. For next generation sequencing, you basically, I have to say it really depends on your protocol that you're using. You will need some kind of pre-amplification step to generate enough material for the library. And I would say, I would probably recommend to use as much as you can, and really try to figure out what amplification method gives you good results. We actually, QIAGEN, we just released recently a single cell library preparation kit that we tested, or we're still testing for analysis of long RNAs from serum and plasma, and that looks pretty good or pretty promising. But it really depends on what method you're using. So it should be enough, but you need some amplification that is powerful enough. It should be enough to give a good representation, I should say. 200 microliters for microRNA and 1 to 4 ml for messenger.
Okay, we have time for one last question. The question is, does use of carrier RNA improve yields?
Yeah, like I have shown you how the linearity of the recovery, and we do not, first of all, the recovery that we do see without carrier is pretty much perfect, so there is no indication that we get lower recoveries when the volume or the amount of sample declines. So that's the first indication that is really not necessary. And also, when we actually add carrier, we do not really see any improvement as far as we tested. So from my experience for RNA, at least in general, I think the carrier helps in precipitation-based protocols, but for spin column-based protocols like RNAsy or whatever, it doesn't seem to be required. I'm actually, sometimes I'm really surprised how far down it remains linear, basically as far down as we can detect the RNA by PCR.
Okay, great. I would like to thank our sponsor, QIAGEN, and Dr. Schlumpberger for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through November 2015. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to all of your colleagues who may have missed today's live event. See you next time. Goodbye.